Hello. Just waiting
Hello.
for a few people to join. All right, we get started. So I'm Peggy Angel. I'm chairing this interest group of New Frontiers in Spatial Omics. And Kristen?
Hi, everyone. I'm Kristen Burnham Johnson.
Yeah, so today we're going to discuss spatial omics and what to do with all that data. So we previously discussed some of the work in brain biology with Carlita Dobrillia, Ramon Sun. We discussed applications in multimodal data analysis for brain with Per Endren's group. Today we have two expert scientists in computational biology and genetics who are going to discuss their projects with us. So Kristen?
Yeah, it's my pleasure to introduce our first speaker. So our first speaker is Dr. Wei-Chu Zhang, and she graduated from KU Leuven in Belgium in 2019 with a PhD in machine learning and multimodal mass spectrometry imaging. And Dr. Zhang was supported by the Flanders Innovation in Entrepreneurship Bakelin PhD grant, which cultivates PhD students' mobility really between academic and industry. And Dr. Zhang leads multiple spatial multi-omic projects for aspect analytics. And the title of her talk is Integration of Multiple Spatial Multi-Omics Modalities and How They Reveal Unique Insight into Molecular Heterogeneity of Prostate Cancer. And with that, go ahead and take it away, Wang-Chu.
Thank you, Kristen, for the introduction. I will start to share my screen. Okay, so I think now you can see my screen.
Looks good.
Okay. So hello everyone, I am Wenqiu Zhang. I am currently a machine learning scientist in aspect analytics. And today I'm very happy to introduce you about recent spatial multiomics work that we have done in prostate cancer, which I did this work in the last 2 years of my PhD, together with aspect analytics and also other academic collaborators. So this work focuses on the linking and integrating spatial multiomics data from serial sections. I think most of you have already heard about spatial multiomics because it's pretty hot now, especially with those development of the technologies. And this can really make us to investigate multiple classes of molecules, for example, RNA, proteins, and metabolites and lipids, and so on. And this can also let us to have a deeper understanding of some complex tissues like cancers. But in this field, there are still a lot of open questions and challenges. For example, different spatial omics data, they might be acquired with different data formats because they were generated from different platforms with different technologies. And they could also have different spatial resolutions. And they might even not coming from the same sections because of the limitations of the experiments. So also, typically in a spatial multiomics study, we require researchers, scientists with different background and expertise. And those scientists and researchers, they might have completely different workflows or work environment. They have different perspectives of the data or the results. So it's really challenging, and I definitely see that we need a reliable, flexible, collaborative spatial multiomics software tool. And today I will just introduce a little bit about how we use this Weave platform, which is a spatial multiomics platform, to do end-to-end spatial multiomics study. So we apply this spatial multiomics pipeline on the human prostate cancer. And because prostate cancer is highly inter- and intratumoral heterogeneous, also its development often involves molecular changes across multiple omics classes. So that makes it a ideal use case for the spatial multi-omics study. So Professor Johan Svinnen and his team from Leuven University, they were leading this spatial multi-omics project in the human prostate cancer. We were involved in this project mainly to help them to link and integrate the data set from serial sections. They collected in total the data set from 8 patients. And for each patient, there are 2 biopsies were collected from both the cancer and normal regions. So for each tissue block, firstly, a pathologist will examine the tissue. And then the serial sections, we did the MALDI imaging to measure the spatial lipidomics data. And this work has been done by Professor Sheng Ellis' group. And from the serial sections, so after the MALDI imaging, we used this tissue for a more detailed pathological annotations. And we're very happy that the experts in human prostate cancer from Weill Cornell, they did very nice annotations on this tissue, as you can see here. They did very detailed annotation. They spent hours per tissue for all 16 tissue samples. And in total, they have around 14 classes over those 16 tissue samples. You will see later in the results that those kind of good or detailed annotations is really important for spatial multi-omics study. You will see much more things linking together. Those pathologists, they also use the annotation tool from the Weave platform. So then we can directly incorporate the annotations to the spatial multi-omics data that we integrate. And in the serial section, we use the Visium platform from 10x Genomics to acquire the spatial transcriptomics data. And this is also from LISCO group from Leuven University. And finally, we collected single nucleus RNA sequencing data for the cell type information. So in total, we have high resolution and detailed annotated microscopy image. And from the same section, we also have spatial lipidomics via MALDI imaging. And we also have spatial transcriptomics data set from the serial section via Visium platform. And finally, we also have cell types data from the neighboring sections. As you can see that the spatial lipidomics and transcriptomics data, they're both high dimensional data sets with different spatial resolution. We use the UMAP to reduce the dimensionality of these 2 modalities to 3. And so we can use this visualization to explain our pipeline easier later. So as you can see that ST, spatial transcriptomics data, and MSI data, they are from the serial sections. So before we can do any advanced data analysis, we have to put these 2 modalities in the same spatial coordinate system. To do that, we have to do a series of co-registrations. So for the ST data and its same section microscopy, we just need to use a space ranger tool from 10x Genomics. But for the MSI and microscopy data, we still need to do the co-registration. And after that, we need to also co-register 2 serial microscopies. And after those things, we have a sort of a registration graph. And based on that, we can directly link MSI data to ST data. So the whole process of the registration is based on the non-rigid algorithm with multiple user-defined landmarks. And this also supported by the Weave platform together. The final result is what we want to see here, that both modalities are now in the same spatial coordinate system. So then you can start to overlay them, visualize them together. But of course, that's not the end yet, because their spatial resolution are still different. For example, for MSI dataset, each pixel covers 30 micrometer, and for each ST spot, it covers around 55 micrometer diameter. So we need to find a way to aggregate multiple MSI pixels to one spot, ST spot. So we use a Gaussian weighting scheme to aggregate multiple MSI pixels into one single representation, and we link that to a corresponding one ST spot. And this is what we're getting in the end, is a matching table. For each matched spot, we have a representation for lipids, transcripts, cell type, annotations, and so on. If you have, for example, spatial proteomics data, then we can add one more column called proteins. And this is really the key step to the spatial multi-omics study, because after this step, you could do much more advanced data analysis, and you can really compare modalities point to point, regions to regions. So as we see, that the real game begins. And so first thing we did was checking this correlation analysis. We try to compare spatial lipidomics data with spatial transcriptomics data. We actually find many interesting negatively and positively correlated lipids and genes expressions. They're not just correlated to each other, they're also correlated to some annotated regions by the pathologist. For example, the PIN region, tumor region, stroma region, and benign region. And we can also generate like heat map to see overall correlations between the lipids and the gene expressions over all 8 patients, 16 samples. Among all of those interesting pairs of lipids and genes, we find one very interesting. So this lipid and this gene, they share very similar spatial distribution. So they're positively spatially correlated in all 8 normal samples, as you can see in those red boxes. They have very similar spatial distributions. But when we start to check all the cancer samples, they are not correlated anymore. And again, you can see it from the red boxes. So here we still don't fully understand what's going on here. The biologists are still checking the results, but we do know some things definitely changed between this lipid and the genes. So the relationship of this lipid and gene expressions is changed across different tissue types. And those kind of significant observations, we can see this because we have this great spatial multi-omics data and integration pipelines. So that's a really interesting result that we have found. And for the last part of the results I want to show today is that spatial multi-omics data analysis can also show the molecular heterogeneity in neoplastic disease areas. So we use NMF, which is a non-negative matrix factorization, to extract features from both MSI and ST modalities. And we find 2 features from both modalities separately that's located and spatially correlated to the PIN region that annotated by the pathologist. The PIN region is in human prostate cancer is quite interesting because it is not cancer nor tumor, but it's a status from normal to tumor. we also find another two interesting features from both modalities separately. Those regions highlighted from the modalities shows the located in the pin region, but more like a sub-region of the pin. And also those regions, they were not specifically or separately annotated by the pathologist. So we are very curious if those regions are still the same as the other regions in the pain. But to be fair, that we did not send the standard FFPE samples to the pathologist for the annotations. So maybe that's also can influence the annotations results. But then we asked them to check those regions again, and they confirmed that those regions actually contain neoplastic gland-like structure. On the other hand, we also check the cell types information. After the spots to convolution and the Weave platform, we can transfer and map the cell type data information to our spatial multiomics matching tables. And we find that in the pink region, there are three main cell types. So tumor cells, club cells, and macrophage cells. So for the club cells, there are not many literatures about this in prostate cancer yet, but we do find that some literatures mention that club cells is related to the formation of prostate cancer, showing the normal cells start to becoming cancer cells. And you can also see this in the MSI and also ST data. And a bit further, we can check which lipid profiles that's spatially correlated with these club cells in a human prostate cancer for the first time. So we can find those interesting lipids and genes that are related to the club cells. And I think some people might ask if there's any AI ML models being used in this study. Unfortunately, not yet, but that's definitely the next step. And there will be a follow-up study about using a deep learning method to do integrative data analysis on this data set. So stay tuned. To conclude today's presentation, I hope I already convinced most of you that spatial multiomics is great. And high quality detailed annotations are so important. There's so many valuable informations there. Definitely use it. And user-friendly, reliable, collaborative spatial multiomics software tools is going to be more and more important in this field. It will bring a lot of great values to the team, and it can make everything much easier. The collaborations, communications is very important in this kind of study. We need to keep talking to each other and learning from each other. And finally, that we are standing in the new era of AI, ML, and spatial multiomics. I can say that we are just in the beginning. If you are interested in this study and want to know more details about experiments and other interesting results, you can check our preprint. And also it's currently under review. Finally, I want to thank all the co-authors that involved in this project. They are clinicians, lab technicians, and bioinformaticians, computer scientists, software developers, biologists. Without them, this study cannot go this far. It also shows the complexity of a spatial multiomics study, and also how important that we need to work together and understand each other. And that's really important for this kind of study. And also, if you have any questions or interested in the study, you can always contact us. And thank you for your time.
Excellent talk, Dr. Zhang. Thank you so much.
Thank you.
And great insights. So what we're going to do is we're going to move to our next speaker, and then we're going to take questions afterwards. And so the types of questions we want are both early career questions, as well as questions about the study. And I forgot to say that Dr. Zhang is joining us from Belgium. Dr. Mantis is joining us from Lisbon. Dr. Burnham Johnson is from the Washington State West Coast of the US, so we're a very international group today. All right, so our second speaker is Dr. Maria Mantis. Maria Mantis obtained her PhD in computational comparative genomics at the University of Edinburgh, specializing in molecular biology, genetics, phylogenetics, and pathway analysis using big data. Dr. Mantis joined Aspect Analytics in 2022, focusing on spatial multi-omics integration studies that involve proteomics, lipidomics, and metabolomics data, as well as spatial transcriptomics and imaging mass cytometry. Many of her current projects also include projects with the Allen House Brain Atlas. And so go ahead and take it away, Dr. Mantis. Thank you.
Thanks. I'm just going to share my screen. Okay, so first of all, thank you so much for having us today. It's a pleasure to be here and to be able to share a little bit about the projects that we've been involved with. So the work that I'm going to be showing here today has been a massive and ongoing collaboration by a lot of different people all over the world. And we're very lucky to be supported by the Michael J. Fox Foundation for Parkinson's Research. So as Peggy said, I am a bioinformatician at Aspect Analytics. I joined the company just a little over a year and a half ago. And since joining, I've been involved in all kinds of spatial omics projects. But my background isn't in spatial biology at all. And in fact, I did my PhD, and I spent most of my time in my PhD doing comparative genomics in bacteria. And so also I'm very happy to be here today delivering my very first talk on mass spectrometry imaging, knowing that this might be a heavily focused proteomics audience makes it a little bit more nerve-wracking to me. So as most of you may know, MSI is a powerful tool for mapping the spatial distribution of lipids and other analytes throughout biological tissues. MSI has many different applications, and lipidomics really stands out as one of them, given that it's relatively easy to detect different classes of lipids. And we all know and appreciate that lipid metabolism plays a vital role in many biological functions and diseases, and it really merits our investigation. For those of you doing data analysis on MSI, you will know that data visualization is based on the mapping of ion intensities across the tissue. And this often follows some type of normalization approach, usually total ion currents or root mean square. And this is completely fine, and what you'll find in most method sections for papers that are out there in the literature. However, there are some problems when it comes to the quantification of your results. And here I separate them into two groups. So first of all, you'll find some constraints when it comes to the interpretation of your findings. And this is because the ion intensities for different analytes are not necessarily proportional to their concentrations. And by this, I mean that if you have two different analytes that are present at identical concentrations, this does not mean that you're going to get the same signal intensity. And in the very specific case of lipids, ionization efficiencies are strongly dependent on the lipid class and the presence of other lipid species, which means that ion intensities can vary by several orders of magnitude depending on the lipid being analyzed. And there's also other things, for example, the variation in the chemical and morphological environments present at each sampling position will really determine the extent of the matrix effect. And this can also lead to variations in ion intensities for a given lipid across the tissue, even if it's present at the same concentration. And this obviously also stems from the fact that in MSI, you have no separation step, meaning that all ions go into the analyzer at the same time. So whilst I feel like we can all appreciate that MSI shows a lot of versatility when it comes to showing region-specific lipid fingerprints and also relative changes across the tissue, it does not often allow for absolute quantification, meaning that you can't often talk about your results in the light of concentration per unit or amounts of analytes per pixel. And even though this is a much bigger problem in MSI, people doing shotgun lipidomics and LCMS will know that you'll sometimes face similar issues. When it comes to our little community, I feel like it became really evident to us doing the study that there are still relatively few studies out there that have a quantitative approach at lipid imaging. And those that do often focus on a single lipid analysis, which we call a targeted analysis. So we really came in here, tried to fill in this gap by developing a method that would allow for the quantification of 13 lipid classes and subclasses that represent almost 200 some composition lipid species, using both MALDI in negative ion mode and MALDI2 in positive ion mode, and pixel-wise normalization of each lipid species across the mouse brain. And for that, we also leverage publicly available resources such as the Allen Mouse Brain Atlas to derive lipid species concentration per brain region across mice that have relevant mutations in Parkinson's disease. So moving on into what we actually did, and bearing in mind that this work involved a lot of different people who were responsible for large chunks of the data analysis and the workflow being presented here today. We, in collaboration with Avanti Polar Lipids, developed a multi-class internal standard mixture that contains either stable isotope labeled or non-endogenous odd-chain species of 13 lipid classes and subclasses. Our mouse brain specimens were provided by our colleagues at Merck in the US, and they gave us 3 different phenotypes. So a wild type and then 2 Parkinsonism-associated phenotypes, both a heterozygous and a homozygous. The workflow was repeated in 2 different labs using mass spectrometers with different resolution powers, and what we did after was pretty standard. So we took 3 biological replicates, we took pulse MALDI H&Es, and here you can see a list of 13 lipid classes that we use in our analysis. And I hope that you can appreciate that the magic really here really is that the splash mix contains a reference peak for each lipid class, meaning that we can then normalize our endogenous lipid signals to the respective class-specific internal standard. So moving on into the actual quantitative MSI, the intensities of our species of interest, be that our target lipid species or internal standards, were extracted through binning of the spectra around the corresponding MZ. And for that, depending on which mass spectrometer we were analyzing data from, the window in ppm that we used for the binning was different. The maximum intensity per pixel within these windows was taken as the ion intensity for that pixel. The images that we go were then normalized by conducting a pixel-wise division of the original MZ image, which is shown here on the left, by its reference image shown here in the middle. And then the resultant pixel values were multiplied by the corresponding internal standard concentration, which we know, to produce the absolute concentration image, which is shown here on the right. And I hope that you can appreciate the difference between our original image shown here and then its normalized counterpart shown here. So before moving on with the rest of our methodology, I really wanted to show you this slide so I could convince you that this method really works and it's quite robust. So what you're looking at here is a correlation analysis between the data that we acquired using the timsTOF and the Orbitrap. And as you can see, there is a pretty strong correlation between the 2 data sets. And we're really excited about this because if you are someone who do any data acquisition or analysis on MSI, you will know that these are usually not reproducible or comparable across mass spectrometers. And this is definitely not true if you want to perform the same experiments in a different lab by different people. So we're very excited about these results. Moving on with our methodology, and from here onwards, this is the part of the methodology and data analysis that I was personally involved with. We did some brain region annotation, and here our methods suffer a little bit of an evolution through time. So initially we had Nathan from Merck going through each brain sample and manually annotating regions of interest using our in-house built digital pathology tool called Annotation Studio. But since then, we moved on to integrate with the Allen Mouse Brain Atlas, and the reason for that was manifold. So first of all, it really allows for more automation. So basically, now Nathan, even though I'm sure that he's very happy that he doesn't have to go through all of the brain regions and have to annotate all of these regions of interest manually, we can derive the regions of interest now directly from AMBA without bothering Nathan at all. We are able to analyze more regions of interest as well. So initially, Nathan was annotating 5 regions of interest, and since integrating with AMBA, we were able to bump this to 12. Now we can do some cross-referencing between our results and other results that are out there that also use the Allen Mouse Brain Atlas, especially if they use the Allen Mouse Brain Atlas with the same AMBA layers. There's a potential to link our data to other data that is available through AMBA, for example, gene expression data, even though we haven't touched on that yet. And there's also potential just for the whole pipeline to be streamlined and faster as there's less people involved. Here is just a list of all of the different brain regions that you can get with AMBA. And I just wanted to say that despite there being all of these benefits from including AMBA in your analysis, there's obviously also some drawbacks. So if you are someone who are thinking about using this resource, you really need to think and plan your experiments ahead, because you need to especially think about cutting your brain samples in depths that are comparable to the AMBA layers. And you need to be mindful of your cutting planes, because if your cutting planes are skewed in any way, then it might mean that your single brain sample might match more than one AMBA layer. So there might be some regions within your brain sample that best align with one AMBA layer, and some regions within your brain sample that best align with a different AMBA layer. And when you have to pick one for whatever process that you're doing downstream, then you're going to be left with a little bit of a problem. And I feel like it goes without saying, but you need to, think ahead that if your region of interest is really reflected in AMBA. As you can see here, most of the regions are pretty big, which is good. So it allows for some margin of error when it comes to the registration approach, for example. But if you're interested in a very specific or a very small region within the brain, then this might not be the best solution for you. Let's see if this works. Okay, so this is a quick, small video of an ion image and all of the regions of interest that are overlaid on top of it. I like to show this because I think it's pretty nice. Moving on with our overlay and integration of our data sets. On the left here, you have the Allen Mouse Brain Atlas. You can derive this data directly from their websites. You can find a Nissl stain that they have, and the accompanying region annotations. For our microscopy data, we got the data in different formats, so we needed to do a little bit of format conversion to get everything in the same page. Regarding the MSI data, we did the standard data processing approaches. We also did our splash normalization that I talked about just now. And we also did some dimensionality reduction to get some images going to use in our registration pipeline. For the registration pipeline, and as Wangshu mentioned already, we used our in-house built landmark-based non-rigid registration pipeline to then register MSI to H&E, and then H&E to the AMBA Nissl stain, which obviously has the corresponding region annotations. And the goal here really is to get a common reference frame where you get the region annotations mapped onto your MSI data. And if you want a quick sneak peek into our registration pipeline, this is it. So on the left, you have the microscopy, and in the middle, we have the AMBA Nissl stain. You need to place 3 markers for your registration panel to show up here on the right. And after that, you're welcome to add as many markers as you feel are necessary for your registration to be good. And I hope that you can see as I add markers that the overlay here on the right improves. And you might ask, how many markers are needed for registration to be deemed good? And this really depends on how close the images that you're registering are to one another. In the case here, you can see that, the images are very different. This is because we're not talking about same section. We're not even talking about serial section. This is a completely different animal. So in my case, for most of these registrations, at least 70 markers were needed, but a lot of times over 100 markers were placed for a good registration. We do offer an automatic registration algorithm that performs pretty well if your images are closely aligned. But in this case, we thought, we could really benefit from doing this work manually so it could get the best registration possible. Okay, so as I said, in the end, we'll get the common reference frame where we have all of the different regions of interest mapped onto the MSI data. And after that, you can extract ion intensities from pixels within each region of interest. And once you have this, you can go wild with your differential expression analysis. You can plot all kinds of plots that you see fits, and you can do all kinds of statistical analysis that, you feel like makes sense for your data. And here I'm just showing one. So here you can see just one of many plots that we have, which shows the mean concentration of a given lipid across different anatomical regions. And, we really like these results because you can actually get absolute quantification per anatomical region, which to our knowledge before the study, it wasn't even available. So this is really nice. And one thing that we actually also did was compare our data here with bulk lipidomics data that is present in the literature out there. And we were very pleased to see that our results were very comparable. Okay, so I feel like most people, or hopefully everyone in the imaging field, are in the imaging field because they like looking at pretty pictures that mean something. So, it's very nice that you can have bar plots with actual concentration values, but I feel like it's really important for you to be able to look at your data to make sense of it. And one of the nice things that Aspect Analytics does is to provide a cloud-based platform where you can do many, many things, but one of them is visualize your data. And so here we are navigating to our multi-sample visualization. On the top, you have your heterozygous samples, and on the bottom, you have your homozygous samples. Where you're seeing these colorful things popping up are the amber regions that you can toggle off. Then you can, toggle these layers on and off, or just make them more transparent to really analyze the quality of registration. So right now, I'm just trying to see whether my microscopy to MSI registration was good, which I hope it was. You can also, in your MSI layer, visualize as many ion images simultaneously as you want, but in this case, we're just looking at one. And then you can toggle your amber regions on top of that and just really look at, how do they differ between mutation types. So now we're looking at the isocortex between heterozygous and homozygous. And we can see, spatially, are they different or not? And I think this is really nice, because you don't have to just, be manually plotting individual ion images and having a look at them and having multiple tabs open in our computer. You can just do this in one go in your platform, which makes data analysis and visualization just much simpler. Okay, so in conclusion, I hope that I was able to convince you today that we were really able to perform quantitative mass spectrometry imaging on almost 200 lipid species across both positive and negative ion mode in a single measurement. Our results revealed sometimes some subtle changes between our data prior and after normalization. And, this sometimes is all that it needs for you to make a new biological insight. So we think this is really relevant. Our data is not only comparable to bulk lipidomics data that is already available in the literature, but we are very happy to know that it's a very robust methodology. That is, we have comparable data across different mass spectrometers, but reproducible data across labs, that are separated by thousands of kilometers from one another. So we're really happy about that. And this methodology can be readily adapted to other MSI modalities, for example, DESI or SIMS. And so we're very happy for you guys to, go out and try for yourselves and see, if you do also get such nice data as we did. And this has really been an ongoing collaboration. And we have, lots of new things coming out soon. We're drafting a manuscript as we speak, so really do keep your eyes peeled for more. And with that, I'd like to finish my presentation, and I'm very happy to take any questions from now onwards. Thank you so much.
Thank you, Maria. What a great talk. Okay, so now we're going to move into the question section of the session. So, Kristen, you want to lead off?
Sure. I think I'm going to start with a general question to both of you. So when did you know you wanted to focus your career on computational biology? And then part two is, when did you decide to focus on spatial multiomics?
I can start, maybe?
Mm-hmm. Yep.
Okay. So my background is engineering. So I did my bachelor for electrical engineering. And then I noticed that my interest is not that. And then it was a bit too late. But then I switched it for my master study for artificial intelligence. And I met two great guys, Mark Klassen and Nico Verbeek. And they introduced me to the MSI world. And I did my thesis on transfer learning in MSI. And after that, I started my PhD and together in the same group with them. And in the early stage of my PhD, I used AI and ML models on ultrasound images and microscopy images for ovary cancer. And that time I noticed that there's still a lot of gaps and a lot of great values that I can bring to the biomedical field based on my expertise in machine learning or computational science. But most of my work or my time in my PhD focuses on MSI. And in the last 2 years of my PhD, I also focus on the spatial multiomics project that I just introduced before. And that really makes me feel the most exciting, but also challenging topic that I want to focus. Yeah.
Great. Thank you so much, Wanchu. What about you, Maria? What made you
So,
focus
yeah.
on computational and then spatial multiomics?
I think it's also bumpy, but completely different from Wangshu. So I'm, a biologist by undergrad. I did my master's on something completely different. So I did it in space physiology. And so I'm someone who, I've spent a lot of time in the lab, all kinds of labs. And even throughout my PhD, I was involved in helping many lecturers with running their practical sessions. So I've done everything in the lab from, DNA and RNA extractions to running QRT PCRs to running gels to doing field work in ecology to, doing physiology lab work, measuring VO2 max and stuff in humans. So I've done all kinds of things, and I feel like I really understood that I wanted to do computational biologist because from the whole part of the experimental process, I was just most engaged with the part of setting up the project. So, everything from experimental design to, what kind of analysis makes sense, what do we need to do to answer a specific question that we have, and then analyzing the data that we got. And the actual part that I enjoyed the least was getting the data. And so it just, it made sense for me starting my PhD to do a PhD that was fully computational, and that's what I did. But it did make for an interesting start of my PhD because I started doing a PhD that was fully programming PhD without knowing how to program, which caused a lot of stress. But when it comes to spatial omics, this is not something that I thought about doing during my PhD. I was finishing my PhD, looking into, going into industry. And I was very lucky that Mark and Nico took a chance on me. I was someone who didn't know spatial biology. I have motivation and I have a broad interest in all kinds of areas within, biology and chemistry to a degree, but I didn't know many of the things that I spoke about today. I didn't know existed when I began working for Aspects. And so, it was an eye-opening thing, and it just really opened many doors to analysis that I didn't know possible, but I really enjoy nowadays. I think it's really relevant work what we do and trying to tackle, very serious and complex diseases such as, cancer and neurodegenerative diseases.
That's fantastic. Yeah, science is a journey, I think, for all of us. We never know where we're going to end up.
Exactly.
One of the things that I thought was really interesting is that many of your studies are international collaborations. And I know when you're handling this big data, there's got to be key things that or resources that you have or think about or ways to get the right data to you. So can you maybe speak about that a little bit about key points in working with a distant team during a project?
Oh, we could talk on forever about this, I'm sure. Um, yeah, I think, one thing that is difficult, but it's very important to get people thinking. And, this is not just at an academic level, it goes all the way to big pharma levels, is that is really important for people to plan their experiments and to be organized in how they manage their data. And you think, oh, it's your PhD students or whatever. No, it's across the field. It happens everywhere. And we really became, aware of this. And that's why, we offer data management, solutions. That's one of our big selling points, to help people really manage their data. With international collaborations, I feel like it's not necessarily, in my opinion, I don't know if Wang Shu disagrees, but I think it's more the difference in expertise than the geographic location. So I think it's harder to get people that are, biologists talking to bioinformaticians or software engineers talking to, pathology experts. If you need to, then to get people, in the US talking to people in Europe or people in Australia. I think, you might have some uncomfortable meetings at uncomfortable times, but in the evening or very early in the morning. But I think the real complications come from communication between people in different fields.
Yeah, I agree most of the things that Maria said. Maybe one thing that I would add is that the regular meetings and updates is very important. That you know what's going on from different groups, and also that my background is not biology, so I have no idea about biology. So I have to have multiple meetings with biologists trying to understand what they're looking at, what they want to do. And I feel like both of us, like all of us, we need to be more patient in this kind of studies. We need to spend time and effort to build this communication bridge between each others. And we have different terms, we have different way of analyzing or perspectives of the data and the results, and that's really takes a lot of effort, but that's all very important and valuable to this kind of project. And I think I was not that patient, but now I'm getting more and more patient. I try to explain to others what I'm doing and what's my next step and why I'm doing this. I think it will make everything much easier for all the other sides. Yeah.
That's great. I think I'm going to pull on that a little bit more because, what about before the data? So as computational biologists, do you guys have any suggestions for the scientists working with you in regards to designing the experiments and acquiring data? Are there special tidbits of information you can give in that regard?
And I think before you start the experiments, think about what you really want to do and what's the real question. Because sometimes we see that people have this tissue block and they do the sectioning, and sometimes they notice that, oh, actually we want to see this modality and this modalities, but they're not serial section, they're too far away from each other, and that's very pity. And I think that especially in the spatial multi-omics domain, serial section is very important. You have to check in detail how far they are. Are we still looking at the same cells for the downstream analysis? And also important that please try best to document the data and experiments properly, and try your best to log all the meta information during experiments, because we received so many different data sets from different groups, different labs, and we have to link them together for the downstream analysis. We spend so many time to trying to figure out which data, which modalities is for which patients from which sections. That's a lot of tedious work. And if everyone in the lab can log their information in detail, that will really help us. The downstream data analysis part will be much more efficient. Our life will be easier. Yeah.
I feel like I can add, so I feel like we can definitely provide help into, how you can run your experiments. Maybe not to the, what reagents you need to get, or how you run such a experiments in the lab per se. But I think, we've seen a lot. We've seen pretty much all kinds of data combinations out there, and data formats out there, and where things go wrong. And I say for myself, but I feel like sometimes we really be nice if you were involved in setting up the projects, because sometimes we're not, if a company reaches us wanting something, this might be data that they have had for years, it might not be something that they're doing then. And planning a project, it's very important. And we feel like people nowadays are really eager to jump into the data acquisition before really thinking what makes sense, not just from everything. Like, do I have the materials that I need? Do I have the time that I need? Do I have the budget that I need? And then running the samples. Is the data that I'm acquiring, is the assets that I'm running the right thing for me to answer my question? Do I have a question? A lot of people come to us with all kinds of data being like, well, we've done all of these very expensive experiments, but we're looking for something interesting, and they don't know what they're doing.
Yes.
Which does make for, because the technology evolves, and with these technologies, it's terabytes of data that we're acquiring. If, for example, for this study that we ran now, we have 12 different brain regions and we have 200 lipids. If you don't know what you're looking for, you're going to spend years just looking for something, and I feel like if you're trying to force a result without a hypothesis, then that's when you get biases, and so we do feel like project planning from start to finish. So from do you have what you need, to what assets do you need to run, to what statistical analysis makes sense to run given the data that you have? Can I derive the right insights from what I just did? Because we do feel like a negative result is a result if you have a hypothesis. And then just bringing everything to a conclusion. I think it's really important, and we do see that lacking sometimes.
Thank
That's
you.
great.
So I have one more question, and then we have two audience questions. We have around 6 minutes left. So I was looking at the annotation studio. So we have the mouse model, and then we have the human tissue. And so I know, Dr. Zhang, you had said that it looked like you had a pathologist that annotated all these pin regions, which I know is so tedious. And then did you move to a pixel classifier like QPath, or was that Annotation Studio? Is that
Yeah, so
particular?
Annotation Studio, they label them
Yeah.
and then they can export the annotations into GeoJSON file.
Yeah.
And then we just use the GeoJSON file for the registration because they're from
Mhm.
the microscopy data. We need to register them and pixel merging for the spatial
Mhm.
spatial multiomics matching tables. So for each spot, we
Mhm.
have annotation from the pathologist.
Okay, thank you. And then for the mouse studies you were comparing those to the AMBA annotations, which I thought was really cool. And I wondered if you had done any three-dimensional studies.
No, but
Like,
we
to take
are
it to the next
starting.
level.
Yeah, we have it's something that it's been in our roadmap for a long time. People have asked us many times, and we are not necessarily for this project. Maybe it's something to think about, but for other projects that we have ongoing right now, we are now starting to develop 3D
Yeah.
visualizations, which
Soon
I'm
we'll
excited
start
for.
building entire organisms. So we'll have the 3D, then we'll we'll get
Exactly.
more and more
Yeah.
complicated. Kristen, do you want to lead off the audience questions?
Yeah, I can. We've talked a lot about serial sections with your guys' talk. And then Robin asked, have you performed mass spectrometry imaging and transcriptomics on the same slide?
No, because we received the dataset from the serial section when we joined the group. But we do know that from SciLife Lab and the PERS lab, they have these new experiments that they can measure both MSI and ST data from the same section. We're also testing our pipeline on their published datasets. Yeah.
Great.
That will be very interesting in the future to move on from the same section. It will have much less challenge for the co-registration, and you also don't need to worry too much about are we still looking at the same cells. So definitely nice directions in the future.
I like that you were combining different modalities. We have a lot of lipidomics imaging we collect on the high-end FTICR instruments, and then we have other data that we collect at very high spatial resolution on like the multi-QTOF type of instruments. So I was really excited to hear that.
Yeah.
Have you done any combination of MALDI and DESI yet? So I know DESI's great for drug
Yes.
mapping.
We have a collaboration with Swansea University in the UK, and they have DESI, AMBA, and MSI. Yes. Well, I think it's I don't know if it's Orbitrap or but it's yeah.
I guess our final audience question is, what are the false discovery rates of the mass spectrometry imaging results? Maybe you can just super briefly in 2 minutes that we have left, talk about where you get those IDs.
Me? Yes. Well, the IDing of the lipids were done by Shane Ellis's lab in Australia. It wasn't something that we were involved with, so I would encourage you to read the paper, and everything is described there, but I'm not so sure about that.
From my project, you can also check the paper. There's a section to describe very in detail how did we do the ID. And it's done by a LISCO group from Leuven University and other collaborators. I personally didn't do that part.
Thank you so much. All right. Well, we're right at the end of our time. So I want to thank both of you so much for preparing this talk and answering some questions about working in these interdisciplinary teams.
Thank you.
Yeah.
Thank you for
Thank
giving
you
us the
both.
opportunity.
Thank you so
Yeah. What
much
an
for
enjoyable
having us.
talk. Thank you.
Thank you. Okay.
All right. Bye.
Bye bye.